lab on the hoof. I have done this before in Egypt when I worked at El Amarna and also at Thebes, just outside Karnak. The Nebulos Gate, which is in modern Luxor. In Egypt, you can't take any soil or sediment out of the country, and particularly that from archaeological excavation. So you have to do all the processing you possibly can in situ on the spot. And this is way back in 1979, I think. So I suppose I had my MPhil a year by then. And I met a Canadian guy called George Hathaway, who's now like a senior court judge for the Ontario judicial system back home. And he was an archaeological scientist in the making then, and we decided we would take all the kit we could find from Canada and from Britain out to and set up in the inspector's office on the site. So we took our own sieve stacks, we took a very rudimentary early computer, we took a rheostat with us so we could adjust it to the current fluctuations so we didn't end up wiping out our data. At the same time, I borrowed from a jeweler in the souk in Luxor, one of those amazing weighing scales, with the little brass weights you put on, so I could weigh all the evidence from the particle size work that I was doing. And I did by hand, like Katerina, we were talking about it this morning. Katerina still teaches her students how to do particle size in the old-fashioned way before lasers were invented, so to speak, which make it much quicker and easier, but perhaps don't give you an idea of the understanding. And so we did, and I still do this really, although it's expanded a bit more recently. We try and do is a suite of physical characterization tests to go with the micromorphological blocks and thin sections, which we'll look at a bit later. Try and always do pH, loss in ignition, magnetic susceptibility, and probably phosphates. And if the phosphates look really good in terms of information return, may well do multi-element analysis, which we talked about the other day on site. And then the other thing we would do if I was particularly involved with sites that were waterlogged or drying out, ones I'd want to do some electrical conductivity or EC, which this meter can do, and redox, because they're very good at telling you about potential preservation conditions and whether the organic components may or may not be surviving despite modern dewatering and so on. And so it's, none of it's rocket science. A lot of the kit is relatively cheap and cheerful, lasts for a very long time in terms of longevity. And in terms of, these meters, they sort of range from about 100 pounds up to maybe 400 pounds. And you can get them for all sorts of things now. And there are even meters, I don't know whether you've ever experimented with them, Katerina, that say they're like probes that you can literally put in the topsoil, and they're supposed to be effectively giving you multi-element analysis. And I've never trusted them, okay? All right? Okay, we've tried them, we've had salesmen try and sell it to us, but I still think it's better to do the wet chemistry, simple wet chemistry lab work. And the other thing that obviously is flavor of the month, which I didn't talk about this morning, is portable XRF. And obviously you could do it in the lab. Petros does a lot of it for on lithic materials. But for example, good case in point, it's about 10 years ago now, we were digging a late Neolithic settlement site on the edge of Durrington Walls, so one of these big embanked and ditched henges just near Stonehenge, a couple of kilometres away. And it had these lovely little rectangular structures about the size, twice the size of this bit of trestle table here. And the archaeological curators there, which was owned by the National Trust, they were so paranoid about me putting my divots, my little sample pockets, into the floors, which were in situ because they were covered by later midden material and then the main henge bank. They didn't want lots of systematic holes all over these sort of chalky rich floors and the central hearth. So I was allowed to take one micromorph block and one small bulk sample next to each hearth, and that was it. Because they said, oh, we've got somebody from the University of Bradford doing PRXRF on a 50 centimeter grid across the whole open area excavation across about 20 structures. I said, okay, but I really think you should for insurance, because we'll never get back here. Never. And of course the results were gobbledygook because they hadn't calibrated the kit properly. And so it's like, no data. And it's a disgrace really, because in Britain, there's, unlike here, there's about half a dozen neolithic settlement sites in all of southern England. It's not like they're every few kilometres like it seems there are here, to do replications. So a good lesson it's better to oversample, as I said, on site, and sub-select out of from that to actually do the work. And one, of course, I often do is a sort of, say you might have assemblies, from this, we'll probably have maybe 50 plus small bulks and maybe 25 micromorphological blocks. I tend to usually try and pick what I think is some of the, or say a dozen of the best samples that are representative of the stratigraphy we've got on site, and process those first to then really see if the information return is good enough to process some or all of the rest of the ones you've actually taken. And of course obviously be able to budget to the excavator or the project, how much these stages will cost. And it is often a useful thing to do on route. So the reason I like to do pH always first is because it's a very good indicator basically of the groundwater and geological terrain's acidity or basicity, alkalinity. And it can also sometimes be a good pointer, because of that, to whether you may or may not have good organic preservation. So, on this site, probably going to be alkaline because there's limestone hill up the top above the site, and it's, as you know, it's pretty dry, most of it. It looks like it's highly oxidizing anyway, so it means that anything that's unfired is probably going to be completely humified, hence those lovely rich dark brown alluvial topsoils we see under the occupation zone that we've seen in the profiles. But another way you can check, and this is particularly where it comes into these waterlogged sites, we'll use electrical conductivity. So if it's in the sort of plus 100 to, 200 to 400 and above, then it's really damaged in terms of humification. If it's getting closer to zero, it's not so bad. And reduction and oxidation, like with pottery, reducing air excluded, oxidized air, aerobic type of oxygenated environment. Redox is very good. So if you get nice negative numbers, particularly 50 to 100 plus, you've most likely had waterlogged air excluded conditions in the past. So that means, usually it means pollen, for example, should be really well preserved, uncharred organic remains should be preserved, maybe even insects, all sorts of things, and textiles, all that kind of thing will show up. And I've now been part of about, 4 major long-term monitoring, hydrological and geochemical monitoring projects in Britain to look at these waterlogged sites that are being threatened by dewatering today. So Starkar, the big Mesolithic site up in Yorkshire, a couple in Cambridgeshire, and also down around Avebury in Wiltshire. And it's been a really important tool. If you get more than 2+ years in one site, we tested it for a decade. So we did before they quarried, whilst they were quarrying, and then after they quarried, and we restored the landscape over a decade to try and amass, what are the effects of dewatering on particularly the paleoenvironmental record. So yeah, pH and redox are really good on that front, and obviously other things like dissolved oxygen as well. So another thing we like to use is loss on ignition, because it gives you a really good idea of the amount of both carbonates and organic matter in the soil system, and you just need a muffle furnace and burn it off at different temperatures over about 4 hours to actually get those results. And again, that can particularly help if you're, say, worrying about in a very deep, thick buried soil sequence where you're maybe getting cumulative stop-start topsoil aggradation, it will often be a good indicator of where the real buried topsoils are in the past. And obviously where there's midden type, lots of organic waste, that kind of thing. And complementary to that is magnetic susceptibility, because that will tell you about not just places that are burnt, but also say where you have, maybe upwellings of groundwater that dry out really quickly, and you get lots of secondary iron formation. It will pick up that kind of thing. It can pick up sometimes the surface of old land surfaces, again where you're having trouble identifying in a homogeneous sequence where the old land surface stops and starts. Obviously it's good at giving you a good idea where there's cremations, say pyres, those sorts of things, but it won't give you the temperature, okay? To get the sort of amount of burning in terms of degrees centigrade, a combination of using thin section micromorphology gives you a fairly good qualitative guesstimate of temperatures, but the real best way is using this FTIR, so Fourier transform infrared spectroscopy, effectively developed by the Israelis, Steve Weissman and the Weissman Institute, where they've been digging these amazing big Bronze Age tell sites on Megiddo, and again setting up a lab on site to do all this kind of thing on the spot as the material literally comes off site. So they have a microscope hooked up to an FTIR piece of kit, which costs $250,000. Nice polarizing microscopes like Katrina's. They do the MagSus, pretty much everything we're going to talk about today, except for making the thin sections, they do on site in a big tent right next to the long-term excavations, which is obviously the ideal on any big field project, but obviously costs shed loads of money, and they have lots of American US dollar extra monies, shall we say, as part of the deal. Yeah. So and David Friesen, who was in my lab for about a decade, he's just got a lectureship in Haifa, was responsible for setting up the same system of FTIR in our lab. And they basically, the Weissman Institute just gave us their library of 2,000 curves, which show us about the degrees of burning in different types of soil materials, so we can actually make sense of things. And Petros, who's here now, is our sort of main in-house expert at the minute on interpreting this kind of thing, because it is actually quite difficult to get your brain around different things. And because basically clays, the more they're heated, the more they become agitated and change their structure and their component parts. And that's what gets picked up in the FTIR. Okay? So obviously we won't be demonstrating that today. And what we will also be doing a quick demo without any measurement is phosphatization. And you can see this lovely one we did from this morning, turning this fabulous slatey blue color. This is the topsoil at core 1 at about 10 centimeters. And basically when you have a colorimeter system, which is sort of a cheap and cheerful version of what multi-element ICP analysis does, effectively you set the colorimeter to wavelength of light bands depending on the element. So for phosphate, in phosphor it's about 425 nanometers, and getting these blue colors, and you basically use the supernatant liquid, which we've already pre-prepared from this morning when it goes clear. Usually filter it through filter paper, add a couple of reagents which get us into the blue spectrum, and then you pass light through it in this colorimeter, and it will actually turn the color of the light, or the light of the color as it were, into a number in terms of percentage phosphorus and phosphate. And particularly it's a very cheap and cheerful way of doing extensive systematic phosphatic surveys over quite wide areas, particularly to do with settlement sites. And usually, of course, it's total phosphate. It doesn't discriminate the inorganic and the organic side of things. So it's often, sometimes you need to do multi-element or do some individual segmented, as it were, inorganic, organic, and total phosphatic work in the lab separately to actually tease out what's coming, say, from organic waste versus bone versus what's inherent in the system, or what what might be contamination by modern fertilizer and the groundwater system. And probably what I forgot to say on site, the reason we always start by sampling the modern topsoils and the sort of weathered surface of the geology, you need to actually be able to establish some background levels. So you might find, for example, where we are now, there's been so much fertilization over the last 20 years that everything's contaminated, polluted in effect by phosphates coming out of the fertilizer, which makes it almost impossible to interpret the archaeological levels as the goes down in the groundwater and gets deposited on the archaeological intact surfaces. So it can be a dangerous thing to use on its own. And of course you can see phosphatization in thin section as well. It shows up as very distinctive look to it. Often it's in combination with clays and iron, often in coatings and intercalations. But sometimes it's in the pores, and it often looks like, imagine if you turn over a nice mushroom and you get those brown gills, think of them being a lovely sort of crimsony orange color. That's the way phosphatization looks like down under polarized light under the microscope. And it stands out at you as being different. And then sometimes the whole matrix will be phosphatized, and we use fluorescent light attached to a polarizing microscope, which obviously we haven't got here with us today, to get an idea of the sort of the wider halo of effect of phosphatization of whichever level you're looking at in grid section. So let's do some basic. We'll do some just basic pH. Do triple readings of everything. Make some notes. And then do one or two phosphate samples, and then we'll swap over to looking at some slides. The first slides Catherine has got from the near adjacent other Neolithic site, just about 3-4 kilometers away to the northeast, I guess it is, I guess.
Yeah, you remember the first day when Raina told you to look at the church tower?
Yeah.
That's the site.
That's right.
Yeah, halfway up there is the site, yeah.
Gorjani, right?
Gorjani, yes.
That's the one. So with the first stage of preparation for pretty much everything to do with small bulk analysis for geochemistry, physical analyses, and multi-element and it's pretty easy relatively here, is first thing is, obviously you let your sample air dry, put it in I'm going to leave the larger lumps out, put it in a pestle mortar and grind it down so we get it all to silt size, and we'll pass through the 1 millimeter sieve easily. And the reason, I'll just do before we start. So the standard sort of set of sieves really for removing the stone fractions. So we want, for soil work, we want the less than 2 millimeter size work for all this work. This will remove the stones or pottery or all the other extraneous stuff that may be in your sample. You can, sometimes I will weigh, it won't count it, but I'll weigh it and give it an estimate of the size range, 1 to 2 centimeters, whatever it is, as part of the record. And if we were doing a soil stack for separating out the sand fractions, effectively near enough the 1 to 2 millimeters is the coarse sand. Lost our washer, so to speak, here. And then half a 500 microns or half a millimeter down to 250 being the medium sand, 250 to 100 being the fine sand, 50 to 100 microns being the very finest sand. And if you start off with for particle size with a known amount of dried pestle and mortared and sieved less than 2 millimeter material, say 40 grams is what you would start off as for particle size analysis. Then I'd usually put it in a beaker, a glass beaker on a hot plate with say 100 ml of distilled water and some hydrogen peroxide to burn off, effectively boil off the organic humic remains that are actually in that system. And so say you might start with 40, you might end up with 35, say, grams left. So that's your your total, and then you obviously dry it out again, and then put it through the sieve stack to remove the sand fraction, weigh what's in each sieve to get the percentage of that 35 grams that actually is still extant. And then you put it, the rest of it through, just to catch everything, through a 63 micron sieve, which effectively is the difference between the finest sands and the silt and the clay fractions. So in this you'll end up with all the sand. Put it in a funnel on top of a 100 millimeter graduated cylinder, distill water again, fill it right up to the top. Obviously use this to dry and use for sieving. And of course the silt and clay component will gradually settle over time in the graduated cylinder. You mix it up for half a minute, or I do anyway, and I use a hydrometer just like if you're making homemade beer to get the specific gravity. And some clever soul, I think probably in the 1950s, worked out that at half a minute, 2 minutes, 7.5 minutes, 30 minutes, 2 hours, 6 hours, that discriminates the coarse, the fine silt, and the clay components. And you convert the readings you get in the hydrometer from this table of magic table through to a percent of, and you end up with your frequency curve of clay, silt, and sand. And that's how I used to do it by hand. And I used to probably do about a dozen a day if I was really very finely timed what I was actually doing. I'd be trying to do other preps in between in the gaps. Sometimes it could be quite a rush, but it was always quite fun and juggling act to do these things, to actually whistle through them. And of course now you probably use 10 grams and they shove it in these laser systems and you get the numbers out the other end, which is kind of cheating really. Cheap and cheerful, but also I've found, I don't know about you, Katrina, but we've used on particularly samples where there's a lot of clays, and we got a lot of aggregation, so it's even though they're very small, sort of pellet-sized, tiny aggregates, they're not being disaggregated properly by the laser. So you get these erroneous high clay readings, which if you compare it to by hand and what you're looking at down the microscope, they don't match at all, kind of thing. So I've actually become quite distrustful often of the laser kit without better preparation. And we particularly found this in the Chilean soils, for example, and the Peruvian ones. And so we've ended up having to re-break the soil samples down again to actually prepare them again for operating in the laser kit. And of course, to me, still, just one last thing, the best check is still in thin section, because you get the relationship, the sand, silt, and clay, all in one field of view.
I'm just wondering, why is it? How does it with this organic material? Why are you burning them out? How would this implicate on the result if you would not do this?
You'll just get a slightly expanded amount, because again, the organic matter is often responsible for getting these micro aggregates, which makes it look like, say, coarse silt or something like that. It makes the particle size not as accurate. Yeah, because it's effectively a binding agent, and it helps create soil structure and so on. So if you don't break it down, then you may get these slightly erroneous results. Yeah. And obviously, if you do loss on ignition, you'll get a check on how much percentage-wise organic matter is in these samples in any case, and also tell you how much carbonate you're burning off as well in the alkaline environment.
After all these basic analysis,
Yeah.
Does it exist like CAN method for elements in soil?
I'm not too sure. CAN.
SEM.
Oh, SEM. Sorry, big point. Yes. Yeah. We tend to do that where we don't understand something else, or that's how I use it anyway. So, for example, if I want to really know the elemental and fabric composition, say, of a substrate that I'm not familiar with, so I used it in southern Spain, say, on these ancient marine marl systems near Almeria, because I could see that all the hill wash and buried soil systems were being weathered out of these ancient marine materials. So I took individual samples for SEM analysis specifically, so I could work out what the weathering complex was.
Yeah.
So yes, it's very useful for that.
Yeah.
In particular. And of course, I didn't talk about this this morning. It's also incredibly useful for clays, because obviously in thin section and so on, you can just say it's clay, and whether it's pure or dusty or dirty, you can't say it's kaolinite. Whereas if you use the SEM, you can actually put a name to the clay type.
Yeah,
That's
I wanted to
that's it.
because of that.
That's
Yeah.
it. That's it.
That's
Yeah.
what I did for some caves. For some cave segments, since you see, the clay minerals in the microscope. So we did SEM.
Yeah. And then the other thing I've done also, just before we start, this is sometimes when I'm looking particularly at the nature of hill wash and what does it take to make the soils in this landscape become on the move. I use a couple of engineering tests called Shear and Plasticity, and effectively you take, with the micromorphological block, you take another one of a known volume, and it's the kind of testing you do if you're making a big highway, when you have to test how much the soil and geological system will take in terms of the weight of the road and the lorries and everything else on it. And basically the shear gives you, how much weight does it take before literally the soil structure just disintegrates and it will move down slope or start to move and be disaggregated. And then the plasticity is how much moisture it takes to make the soil mobile.
What is it called?
Plasticity, so like plasticine. With
No. The type of analysis.
Oh, shear testing and plasticity testing.
Yeah.
Should I write it? I'll write it down.
Yeah, please.
Shear ti je posmak.
Okay.
Ja mislim, ako ne znam kako se kaže na srpskom, sorry. Shear je posmak, je li tako?
And engineering departments do it as a matter of course. Big development projects. Yeah. So for example, in terms of southern Spain example, we found that it only took about 40 to 50% saturation for the soil to become highly mobile, and they're very high, sort of an alkaline silt. All very close to what we have as the topsoil here, and it might well be similar in why we have so much material accumulating. It moves incredibly easily, partially because of the particle size, but also because there's very little structure to bind things together. So it becomes highly mobile in moisture from rainstorms, and with only a few degrees of slope. You don't need much slope to make silt mobile. And then in the opposite, of course, when it's really dry, it's also the first particle to move in the wind.
Yeah.
Right. So if you want to start to grind that up, that would be great. No need to be too gentle. I tend to use a great big one, great big kitchen where you can really whack it. That's the economy of one brought by Kathleen. I must get something like this. It's really bad. Is it a kitchenware type one, is it? You got a lab one.
I'm gonna cut this out anyway, parts of it. Of course, then I now need to service the sprinklers for the court. But as an Englishman, I'm sure you understand.
Of course.
And you prefer the turf.
Yeah.
To
But why are they watering it in the height of the sun?
Maybe someone is like.
Maybe so.
I wouldn't complain because that same guy, who's leading them, is roasting our pig. So
I can't do it.
and he told us when we have pause we can go there and see the roast. He said later.
Probably good enough.
Tomanta, ne brineš, bit će tvoj video, ali ćemo prekid zvukova na naših tračanja, znaš.
Dump it in there. And we should have a like a paint shaker type thing for the sieves, is what you can use. But this is good enough at this stage, really.
I uzmite si soka I vode, molim vas, donesem vam svima.
Because this amount should be more than enough what we need. And these are the pots for magnetic susceptibility analysis, but the reason I brought them, I thought, I don't have a weighing scale, obviously. I thought this usually takes about 10 grams of soil. I thought it was a good way of cheating approximately to more or less get the right sort of size amount. So if we just dump this lot out, and if you want to fill that to the top, but not overfill it, just level with the top.
I can do it just like
Yes,
this.
you can do it like that, but
I
I often pour it in, but yeah, how are you going to do it?
Not to the top, right?
Yeah.
Like this? Or more?
Right, to the brim.
To the
brim.
okay.
To the brim.
Why is not going? It's not anymore.
It's
not enough.
It'll do. Plunk it in there.
Like this?
Yeah, just chuck it in. And do the next one.
Okay.
Next one. Oh, give it to somebody else to do. Let somebody else do it.
Okay.
Yep. And we're going to add. And the protocols for all this, they are mentioned in my book, but I can send grid sheets of the methodology. I'll get them all put on site. So we add about 25 mil of, should be distilled water, but hey, we don't have distilled water. And we already tested the pH of this water this morning to know what the error is going to be. So if somebody wants to write it down, this is a pH of 7.47, which is close enough. And you say, well, how do we
write this down? This just one. This.
Oh no!
No, you don't.
Just say
we
bore.
don't
Well, just say we do, but we just say for borehole 1, the distilled water we're using from the bottle, or the water we're using from the bottle, has a pH of 7.47. And normally, what we would do to calibrate the machine, you can get these sachets of pre-prepared pH at the right level, so at 4, 7, and 10, for example. So here I would have probably picked a sachet of 7, so you calibrate the kit to 7, so it knows exactly where it is, so you'll then be more or less accurate when you come to test this material. So if you want to write down, so 120 to 80 to 120, and then 120 to 140, the next one. That being the last one. And that will be my third one.
We don't have yet. Ty ne nosiš uvek dvakevica tamo?
Možno, če nemam še koga, to sem
A to je tvoje?
vreći. Ja,
Ti bom vzela eno.
to razumem, lahko kemik. A je to dosti?
Ja.
Okay, lovely. You want to shove it through the stack? Let's check it in. Thing like that. Go back and I want to pass the last back again.
pot if you can. Yeah, I tend to pour it in with
Yes,
it's quicker.
It's
Don't worry about it, Skelly. That's not the end of it. Chuck it in a small beaker. And what I would normally do, I just chuck it in, but what I would normally do before I do the gauge without, because I use these parts of mag sus machine, so I do mag sus, do the grinding and sieving, then mag sus, then pH, then phosphate. It's the order I would do it in, so you don't waste the precious amount, you just sort it out.
And third one, right?
Please, yeah, thanks.
Там и стресса пару.
Thank you.
Я съм си създавала за магистър, ще мекам.
Абе?
Is it good enough?
That's I'm sure it's pretty good enough. Always grind small. Chuck it in.
Yeah, we might need to actually grind some more.
Grind some.
Yeah, please grind some more. It's not going to be enough. Not quite enough, I don't think.
To jest nasza.
Yes.
Ja. This one goes easier.
Yeah, it's looking good. That's looking good.
This one is not so easy.
Great.
Good.
Clean up a whole lot. Super. Thank you. Hopefully just enough. Magic.
And what is the volume of this small cylinder?
It's, I think it's 10 milliliters. I think. I mean, this is just cheating. You should really weigh it out on a weighing scale, really. You know. But, I just thought this is the best way of doing it under our primitive circumstances, as it were.
And if you weigh it, you take how many grams?
10. So yeah, it's a ratio of 10 grams to 25 milliliters of distilled water is what we want for our concoction. Okay, so.
And when you prepare samples like this, don't you worry about the contamination of the samples?
No. Not really, no. Because, I mean, I guess you'll end up doing enough that if there is real contamination, you'll start to realize that something's amiss.
Mm-hmm.
Yeah. And of course you have
There's several checks really. I mean, again, largely through micromorphological work, you'll soon start to see if there's various things happening that are making it seem contaminated, polluted, whatever. Yeah. Yeah. But yeah, the trick is replication. It really is the way around that. And then sometimes where right, I've worked at one of these big waterlogged sites we worked at years ago in Peterborough. It had tertiary sewage settling beds over the whole top of the site. So it was pointless doing any geochemistry multi-element stuff because the whole place was absolutely full of heavy metals and God knows what all, settling out of the sewage. So you just abandon it sometimes. But yeah, it's always good to ask several generations of landowners, if they're still alive, what's actually happened on this land to help avoid such problems. Yeah. Okay, so we don't have something really to mix things up, so
like for example in our area where there was First World War and the soil is full of parts of bombs and so on. Yeah.
Not so good.
It really is.
That's ordnance is another big problem, isn't it? Yeah, not so good. Out of them. Okay, if somebody wants to. So what I tend to do is, I don't have any paper towel, but use that. Blow on it. I usually put it on its side to get more depth for the probe. Count to 10. Agitate it. Count to 10. So if somebody wants to write this down, first 7.21, then count to 10 again. Again, 7.21, pretty consistent. 7.19, there we are. And so you get the three results and average them, so you could say 7.2, I guess, easily enough. Anyway, you guys should do this, not me. Sorry. So if one of you wants to do the next one, that'd be great. So yeah, just take that off and just clean it a bit and blow through it, and there we are.
Thank you.
That's it.
7.14. Uh, 7.10. 7.01.
Okay. So looking at it, the pH has actually dropped a bit because we probably need to take that that 0.47 off of each of these, so it's around 6.8. So it's dropped from about 7.2 in the topsoil that we had this morning down to 6.8, and that's quite normal to see that sort of thing. It might start dropping for a bit now, and then as we get close to the substrate, it should go back up again. If any, as you get closer to the alkaline groundwater. Right, so yeah, please have a go. Just using the end of the paintbrush to stir it up.
Bilo je 1.4.
1.4?
Da.
Ja, to je bilo. Ne, nije to. Ja. Hvala.
Ampak zmeraj prihajal voda. Torej se bo pač to potem upočeval pri operaciji.
7.50.
50, okay, so it's gone up. So yeah, that's getting towards the bottom of the profile.
$7.55. $7.58.
Great. Okay, so you just want to dump the probe back in the water just to clean it off a bit. And just as a case in point, we can just do the electrical conductivity of this stuff.
Yeah.
Probably meaning 24.7, kind of what you expect, I guess. Not terribly useful reading one way or the other. And it's pretty close in our soil samples as well, 26, 25, 26. Doesn't really help us very much.
And this is in amperes?
That's electrical conductivity in millivolts, so MV, small m, big V. And it's always a hard one to describe, but essentially it's, I guess, a measure of the ability of the soil to attract cations and anions. So it's a reflection of the soil's ability to enable reactions in the soil to happen. So, but on these dryland sites, it's kind of meaningless numbers, really. You really need a wetland site to have some real meaning, to be honest. Yeah. Okay, brilliant. So while we're here, we've got stuff that's nicely settled out of suspension. Normally I would filter through a cone into a beaker the supernatant liquid left over from pH into one of these little vials with a colorimeter for doing pH, or you can set up the machine to do pH, iron, manganese, calcium, pretty much any element you like, kind of thing. But we'll just do this one as an experiment. And you would normally put the this supernatant liquid in the colorimeter to zero the colorimeter. Then you'll add the reagents, which is basically molybdenum blue. So 10 drops of this stuff. Again, all this stuff is off the shelf. This is always a hard thing to get right without putting in too much. And there's a tiny amount of other reagents. So I mean, these, when you're doing something like phosphate analysis, it's less than probably 25 pence per sample in terms of these reagents to actually do it, use it. Again, I can't remember the exact names of what this stuff is, but I can tell you. But effectively makes it go this nice blue color that we saw in the topsoil example here. And then you shove it back in the colorimeter, and you already see it's getting a little bit of light blue, not very much, a little bit. We'll leave it to stand in the, it should be less than the modern topsoil, but yeah, you're beginning to get a blue, move it away from it, a blue hue, just ever so slightly now. So it'd be quite a low reading for total phosphate. So yeah, a magnetometer, sorry, a colorimeter to buy is probably about £1,000, and a bunch of these, you get reagents for all sorts of things. It's a lot slower than obviously doing ICP analysis in a big commercial lab, but it's good for doing, you know, small assessment type work and suggesting whether it's worthwhile doing proper geochemical work on a systematic scale, both vertical and horizontally, spatially, across the site. Okay. Right. So I think we could probably swap over to doing some sections.